All right, we came up to our final topic in our lecture series for Unit 2A, and that is enzymes. And what role do enzymes play? So back, what do we know about enzymes? Enzymes are proteins, all right? We know that enzymes are a function of the proteins. Now, a couple of things that we want to kind of have in our back pocket as we go into this unit. A couple of things. When we talk about proteins, number one, the shape of the protein determines the function, all right? This is going to come into play a little bit later here, but if we have this one rule already down, this will make sense as we go along. So the shape of the protein determines the shape. Now, what determines the shape? The sequence / order of the amino acids determines the shape. And we're going to dive into these two things a lot more. Today, this one for sure, but this one we're going to hit, especially when we get into protein synthesis. So keep this in your back pocket for right now. But let's kind of dive in. So enzymes are something that is essential for life. And the reason being, well, enzymes are essential because they allow for a lot of the chemical reactions to not need as much energy for the reaction to occur. It makes things a lot more efficient. And so if things are more efficient, more things can get done. Our organism can get a little bit more complex. So let's take a couple of questions here. So we want to look at this first question here. How do we speed up chemical reactions? So we know chemical reactions are where there's any breaking or forming of bonds, all right? So chemical reaction would be like H2 plus O2 yields, H2O, and we'll balance everything right here, okay? Now, this would be a chemical reaction. We formed a new molecule. Now, question is, how do we speed things up? The faster, more efficient, the more complex that organism can be. So, what are some ways we can speed up chemical reactions? Well, increase the temperature. Now, let's ask the question why. All right, why would speeding up the temperature or increasing the temperature increase the chemical reactions? Well, temperature or heat will add kinetic energy, making it easier for the reaction to occur. All right. So warming things up, and we kind of saw this in the properties of water lab. When we added the sugar cube into the hot water, it dissolved a lot faster. And that's because that kinetic energy, that movement of those water molecules helps dissolve that substance even more. Now, catalyst, adding an enzyme. Now, why would an enzyme be important in this case? Well, the big reason is that enzymes, aka catalysts, what they do is they speed up chemical reactions by lowering the amount of energy needed. And so we're going to explain that here in just a moment. Now, we can do two other things as well. We can increase the amount of reactants. So if there's more reactants available, the more opportunities that this enzyme that's working here that can bring those reactants together to form our products. Now, the last one, this is a play off a lab that honors biology students did, in which we looked at surface area, or how much contact does a molecule or a solid have to the outside environment. So here's the deal. We can have something that has the same volume between the two. However, based on the shape, we can see that if we have a little bit more different of shape, we can get more surface area so that the enzyme, the environment can go in and do what it needs to do to speed up chemical reactions. So let's focus in on the enzymes. So again, go ahead and highlight this because it is important. When we talk about enzymes, we're talking about proteins, and they act as chemical catalysts in which they speed up the chemical reaction. Now, I'm going to add this little bit right here. They do this. How do they speed up the reaction? They lower the activation energy. We'll talk about that here in just a moment. Now, why are enzymes important? This would be a good short answer question for a test right here. And why are they important? Well, here's the deal. A lot of the reactions that we need in our daily lives require the use of energy. And so if we are able to reduce the amount of energy needed for these reactions to occur, life can be able to expand. Without these enzymes, these reactions, like say hydrogen peroxide, without the use of an enzyme, would have a hard time breaking down into water and oxygen gas. Because here's the deal, this can be poison, okay, if it builds up into your system here, okay? And so we want to make sure our bodies are breaking that down into harmless water and harmless oxygen, okay? This can't occur unless we have this enzyme here to make the process easier, to not have to give as much energy. So let's go ahead and label the basic enzyme that you see here. So in your notes here, this would be object Y, all right? So this is the substrate. This is what's being added to the enzyme. Now, one thing to know about the substrate will be the reactant, all right? So anytime we talk about enzymatic reactions, the substrate is what's going into the reaction. Now we have the enzyme itself, that's going to be the X. But I want you to take note, add this little part right here, okay? This is the active site. The active site, let me kind of put it right here. So the active site is where the substrate binds onto, but there's a key thing that you need to know about the active site. It is specific to the type of substrate. Meaning that, let's say this enzyme's involved with breaking down this deadly hydrogen peroxide into water and oxygen. Well, this enzyme, the enzyme is called a catalase enzyme, and this enzyme will only bind onto hydrogen peroxide. So it will not grab onto anything else. Its only goal is to break down oxygen peroxide into its form once it's done. So if we were going to kind of add on to the picture here, the substrate and the enzyme would combine. Now, this is a question you might have is, what is the enzyme actually doing in this case? Well, the enzyme is doing a few things, but one thing it's doing is causing a little bit of a shift in the substrate compound. We're making the atoms come closer to one another, and when these atoms come closer to one another, well, that causes tension. It causes repulsion. And so as a end up resulting is that because of that extra pressure on those atoms, they're easier to break apart. And so as a result, when my enzyme is done, I'll end up having my products, water and oxygen. So in your note packets, these would be labeled Z products. Now, something to note that's really important that I want you to add to your picture right underneath the Z part, okay? It's this. I'm going to put it in red because it's so important. The enzyme does not get used up. And that's one of the cool things about enzymes is that no matter your enzyme reaction is going to be controlled by the amount of reactants. So the more reactants you have, as long as there are enzymes present, that reaction's going to occur as a result of it. Picture here, we talked about how this binding site, this active site, has a specific type of site. All right, so let's talk about this activation energy. So activation energy, so we would be on page 8 of your notes here. And so let's go ahead and write this down. Activation energy is the energy, is the energy needed to start a reaction. Go ahead and put that down, and let's take a look at this in our notes. So feel free to pause for a second, write down this definition, and let's take a look at this graph with a little bit more gusto here, okay? So let's just kind of get a lay of the land here on our graph. So on my y-axis here, we have the amount of energy. So this would be low energy, and this up here would be high energy. Over here, this is my reaction. So here I'm going to have my reactants. In this case, we're having CO2 and water. And then over here will be my products. So this would be kind of over time if we're looking at it. So we kind of take a look. CO2 and water are pretty stable, okay? We want them to break apart and then combine into this carbonate type of ion, CO3H2. So by itself, this would take a lot of energy to get this to occur. But, so if we take a look here, this first line right here, this represents how much energy it would have to take for that reaction to occur without an enzyme. And the key thing is with this is that it's going to require a lot of energy, okay? And to have this happen over and over and over again, that's just going to exhaust our energy supply. That's going to require us to have to find more food. That's just going to be a problem here. So life found a way, as it always does, to where we have this opportunity to use this enzyme. And take a look here. With the enzyme itself, we have reduced the amount of energy. We have decreased this amount of energy because of that enzyme. And so as a result, less energy is needed because of the enzyme. And that's really the main gist of this is that enzymes just lowers the amount of energy needed for that reaction to occur. All right. Make sure you're able to understand this graph and be able to interpret and explain what this graph is being able to tell us. Straight. Now, again, when we're talking about lowering the activation energy, just like everybody's probably done once, you've rode your bike up the hill. Well, imagine we switched out this regular bike with an electronic bike, okay? That gives you assistance, a little bit of power to be able to get up that hill. Well, with that enzyme, you're able to then speed up and allow that reaction to occur. Now, let's answer this question right here. And this is a question 6 in your notes right here. What happens to the enzymes during the reaction? Okay. Well, here's the deal. The enzyme will be present at the end. What I'm saying here is that the enzyme does not get used up. It continues to work. As long as those reactants are available, the enzyme's going to do what it needs to do. Okay, so An enzyme will not work for every reaction, okay? Like I told you in the so this plays off of that. And so we often call enzymes a lock and key, to use an analogy, because again, you're going to have one enzyme per one substrate, all right? You're going to have one enzyme that's going to be looking for only hydrogen peroxide to break down into water and oxygen. But it cannot bind on to, let's say, a disaccharide and break them off into two monosaccharides. And so the big thing here is that, again, a specific enzyme works with a specific substrate. That is the key thing there, okay? So this is highlight worthy right here. A specific enzymes work with specific substrates. That's our main idea for the lock and key method. And then this answers that question for 6 right here. Enzymes are reusable. All right. Now, like I said again, the enzymes are reusable. So here we have our substrate, we have our disaccharide. It latches onto the enzyme, and with the use of water, breaks them apart, and we have our product here. However, our enzyme is still ready to go and find another disaccharide. Now, how can you recognize an enzyme? All right, well, remember when we talked about carbohydrates that we could recognize any sugars with an OSE. Well, we can do the same thing for enzymes. Helicase, amylase, sucrase, polymerase, these all have the suffix ase, and that always indicates that you're working with an enzyme. All right, now, we're going to hit this a little bit more later on here. However, one thing to note, let's go back to our main idea that we had earlier. The shape of the protein determines the function. So with that in mind, are there conditions in the environment that could possibly alter the structure of the DNA? And that is absolutely the case. So anytime that these enzymes get exposed to a extreme environment, things like high temperature or low or high pH acids and bases, then what we see happens is that enzyme, because again, proteins fold into their particular shape, when they have these extreme forces like in temperature and also pH, it causes that protein to become misshapen, okay? Kind of unravels, if you will. And that unraveling then changes the shape, and so what ends up happening is it won't work as a result of it, or it won't work usually as well. And so the technical term we use for this is denaturation, okay? And that's any time the DNA, or not the DNA, I'm sorry, when the enzymes are damaged in any way by environmental factors, okay? So you can kind of see this again. Same thing here. Here's our normal protein, but extreme temp or pH, and we get a denatured protein. Does not work then. All right. And so this concludes our lecture on enzymes. Be sure to be able to look at a activation energy graph of an enzyme and explain what is going on with that particular scenario.